Okay, so review take 4. Did this yesterday. Okay, so when we're looking at this, we're studying heredity and genetics. Heredity is the study of passing of traits from one generation to the next. So passing traits one gen to next. Genetics is studying kind of how the DNA affects that. Okay? Genetics studies the DNA and how it can impact that. So genetics and heredity, or the DNA and how that DNA can pass on from one generation to the next. Okay? The biggest thing that it all revolves around is this strand. Okay? This strand of DNA. We've talked about the fact that the ladder is twisted, and twisted really, 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 really tightly. Okay? We call that a double helix. Okay? Our DNA, human DNA, most all other advanced organisms' DNA is in a strand. One single strand, this whole piece, is what we call a chromosome. CHROMOSOME. So that whole thing, one piece of DNA, is called the chromosome. One piece of DNA, one section of DNA may be better off. One section of DNA is what we call a gene. Okay? And every single gene out there has different possibilities. Actually, I shouldn't say that. Not every single gene. Some genes are just the way they are, and they're directions on how a cell should work, or maybe how organelles work, or whatever the case may be. However, many genes have different possibilities. Each of those different possibilities we call an allele. A-L-L-E-L-E-S, because my penmanship is terrible. So on this strand of DNA that we're currently looking at, we end up with 1, 2, 3 genes. Each different gene has a different possibility. The gene that I have on top is for if you're right-handed or left-handed. So there are 2 alleles there, a right-handed allele or a left-handed allele. And so the combination of how all these different bases are ordered will tell us that. Okay? And, you know, the next one could be hair color. The next gene could be toe length, or how fast you grow, or whatever the case may be. There's a lot of different possibilities for every gene. Anybody have any questions right now? So to repeat, one strand of DNA is called a chromosome. One chunk of DNA is called a gene. The different possibilities of each gene are called alleles. Everybody good with this so far? Okay. The person who is what we call the father of genetics is Gregor Mendel. Gregor Mendel studied peas in a monastery garden. And he studied the shape of the peas, the color of the peas, the flower color, the height, all these different possibilities. And he came up with a bunch of numbers. And he noticed that we see certain things 3 times compared to certain things 1 time. And because of that, he called them dominant and recessive. Dominant are things that we see more often. Recessive are things that we see less often. And jumping ahead, we know that we get our genes from mom and dad. So if one of them gives you a dominant gene and one gives you a recessive gene, the dominant gene will most likely show. Right? You could have the recessive gene, but if it's paired with a dominant, then you will only show the dominant gene. The recessive gene is with you. That means you're a carrier of it, but you don't show it. Meaning, if you carry it, you can still pass it on to your kids down the road or the organism's kids. In genetics, we like to trace and we like to follow this. And we like to show it. The dominant trait we show with capital letters. Typically, letters coincide with like something you're talking about. Okay? So a lot of times what you'll do, if it's right-handed, you'll use a capital R for right and a lowercase r for left because that's the recessive. So you want to keep those letters the same. Okay? So capital is dominant, lowercase is recessive. So I'm not going to do that. When we study genetics, we also like to predict outcomes. We like to show, hey, what can we get if we do this? Right? And I'm going to talk about this later on. You guys heard me talk about it more yesterday. But in all farm animals and all domesticated cats and domesticated dogs and all these animals, we like to predict what we're going to get because we want certain outcomes. So the Punnett square can give us that ability. So if we want to predict, let's say that dad is right-handed but carries a left-handed trait. Here's dad. Let's say mom is left-handed. Okay, so we complete a Punnett square and we figure out what's the probability our kids are going to be right-handed or left-handed. All right, so the R's go straight down, or I'm sorry, I shouldn't say it like that. That letter from the top left goes straight down. This letter from the top right goes straight down. These go over to the right. All right, so this would be capital R, lowercase, 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 or capital, lowercase, and lowercase, lowercase. So here we'd have a 50% probability of having a right-handed kid, 50% probability of having a left-handed kid. Everybody with me on that? The two on the left are right-handed, however, they carry the left-handed trait. Everybody with me on this right now? So looking at this other Punnett square that I did, let's see if I can just erase part of this. We'll just see. Oh, that ain't gonna work. Okay. Back. All right. So I crossed a capital here. We call this heterozygous. So that heterozygous with another heterozygous, meaning we have a dominant and a recessive. And what we ended up with was a dominant and a dominant. We call that homozygous dominant. Two heterozygous. Whoops, I don't want to do that. And then the last one is pure recessive. Going back to what we talked about a second ago, with the 3-to-1 ratio. 3 dominants, 1, 2, and 3. If you're not looking, that gets cloudy. 1, 2, and 3 dominants to the 1 recessive. Everybody with me on that? That's how we end up getting really a 3-to-1 ratio most of the time. And this is kind of what I started with at the beginning. So, zooming in on this picture here, again, we're going back to a DNA strand. This DNA strand, which I probably should have talked about first, has color coding going on. All right, so I want you to know the different parts of this, okay? So the red, in this case, would be, shout it out if you know it. That'd be sugar. What would the blue be? Phosphate. So that makes up the sugar phosphate backbone. All right, and then orange and green typically are paired together and represent cytosine and guanine. I'm just going to write C and G. If you don't know those, look them up. Cytosine and guanine. Cytosine and guanine are paired together. The other two are going to be adenine and thymine. And those pair together. Okay, let me just back up. You guys got those? Everybody got that? Okay, I'm going to just get rid of all that writing. Okay, what I've drawn in here is this orange circle shows a sugar, a phosphate, and 1 base pair. We call that sugar, phosphate, and base pair a nucleotide. All right, so we have 4 different possible nucleotides, and they're actually brought in like that. Those nucleotides are brought in together, and that's how DNA is built. The last thing I'm going to talk about with this picture is that when we talked a little bit about this, not a lot, but there are groups of 3 that are built together. And we call those groups of 3. Anybody remember?
Base pairs?
Nope, not base pair. It's called a
Codon.
And you guys did the DNA messaging. And so you looked at those sets of 3, and that told us a picture. That told us a story. And that's exactly how DNA works. All of these codons in a row, here's 3, here's 3, 3 more, give specific directions in a specific order on how to build stuff. So it says, okay, bring this protein in. Bring this protein in. Bring this protein in. And so that's how DNA works and how the information is passed on. So each different sequence of 3 represents a different protein or a different direction to do. All right, we good with that?